is working as a technical partner with Jammu and Kashmir Pollution Control Board under the National Clean Air Program, and as a member of Indian National Young Academy of Sciences, INSA, Dr. Yadav is actively contributing in promotion of science amongst the various strata of the society through science camp, remote area lectures, conferences, and other forms of outreach. Over to you, ma'am.
Thank you so much, Rizana, for the nice introduction. Good morning, one and all. And I once again welcome you all to the third day of this workshop. I also welcome our chair for this session, Professor Vijay. So I'm going to talk about aerosol, biosphere, climate interactions and Indian perspective. So my talk was actually scheduled yesterday, and there are some benefits also of talking in the last, and there are some disadvantages also. So I had to remove so many slides because already Professor Pandithirai, Professor Vijay, Professor Sachin, they have shown what I was about to show. But it is also a benefit because now the stage is all set. We know everything almost. So I can go quickly. And so our group focuses on aerosol air quality and climate processes. We work on INP, bioaerosols, source tracers, and source apportionment. So aerosols in news always, and it is because of pollution. So we work in Jammu and Kashmir, and there, if we compare from, say, JNU or from IIT Madras or from University of Hyderabad, there people are more concerned about pollution, and we are also working as technical partner with the Jammu and Kashmir Pollution Control Board. So, but now, as I already said, that the knowledge is increasing. So air pollution is a major challenge in India, and the National Clean Air Program has been launched to work on this. So when we say why aerosols, so many scholars also have talked about like health effects, outside visibility, atmospheric chemistry, Earth's radiation budget, etc. So overall in this room we are sitting to understand the implications of aerosols on human health, climate and overall ecosystem. So when we talk of aerosols, there are different size fractions, what we want to study. Also I want to share with you that my talk is going to be focused more on offline measurements because we do measurements, and offline we do sampling and then offline laboratory analysis. And you can see here different size fractions, size segregated sampling, and whatever you want to do, you have to first decide which size fraction you are targeting according to your objective. And Aerosol Research Group at Central University of Jammu is specializing in offline chemical characterization, as I just said. We are evolving in investigation of bioaerosols. I say this because I'm not a biologist, but now we are teaming up with the best microbiologists of the India and world. So we are improving on this aspect. We are also developing facility to study INP for INP immersion mode measurements, and serving as technical partner to JKPCC, and we are working with policymakers, academicians and local people for capacity building in Union Territory of Jammu and Kashmir. So this is one of the very important thrust area of our group because we are sitting in Jammu and Kashmir, and this is our duty to reach out to the local people, and our university is also in the remote area, so we invite people from schools and colleges, students, and as a NIAS member also we promote all of this to create awareness on aerosols and atmospheric processes. So aerosols are complex, this we know physically, chemically, biologically, and this is a simple cycle, but this is not that simple as it is visible here. So I think all of you must have seen this diagram by Whitby and Cantrell, which shows about like how different processes are happening, and removal processes are there in the atmosphere, making aerosols really, really complex. So when we talk of composition of aerosols, broadly we say carbonaceous elements, water-soluble ionic species. So our research focus on study of physical, chemical, biological and isolating properties of ambient aerosols. So basically these are the three core research areas we are focusing at present on, and first is chemical characterization of ambient aerosols and source apportionment studies. So actually I started my work from the doctoral time on chemical characterization of aerosols, and this is a small facility which has come up in Central University of Jammu. It is called Himalayan Aerosol Research Instrumentation Facility, and as I said, from doctoral research we started working on quantitative estimation of elements in carbonaceous species. So when we say chemical characterization, I want to clarify over here that so many people take it very lightly, right? Chemical characterization. If you ask for the research proposal for the funding, chemical characterization, oh, it's very simple, it's this and that. But when you go to that region, you realize that even the simple baseline data is not available. We don't have even a single ethylometer in whole of Jammu. When we started working, so during the doctoral time, I worked in JNU, we had a time series analysis, this and that. When I came to Jammu, I got to know we don't have PM 2.5 concentration. We don't have even 1 year sampling. So that's why we had to start from scratch, and our university was new, so lots was there to do. But I'm really happy that we are on the right track, I believe. So going back to 2007 when I started my doctoral work, so we started working on chemical composition, and then the major thrust area, 2 years of my PhD tenure, I actually spent on understanding the GCMS data. Because when you get the GCMS data, aerosol is complex, right? So many people work, if you talk to the nanomaterial people and materials group and all, they know what compound they want to do, they'll just put and they'll do. Our matrix is very complex. And to get the good information from that complex matrix is a challenge, right? So that's why you will be surprised when I started working on the doctoral thing and I saw that what source traces organic analysis is happening. So that was a time when Advanced Instrumentation Research Facility came up in JNU. You must be knowing that it is a good facility at present in JNU, and TDGCMS was there. And I got good time to work and play with the instrument. And that time I found that there were only 2-3 studies. I remember Chaudhary et al., Fu et al. And all. And you'll be surprised to know that the analysis was done in foreign laboratories. That means we were not having the GCMS data from India. So it was a surprising fact for me. So, and then whatever the data was present, it was due collected, right? So number of samples is very important. So all of this motivated us to target this, and we started working on non-polars, we standardized method on thermal desorption, and these are some of the results. I will not get into the details. We also studied elemental composition. So this was the first 4-year study in Delhi we conducted from 2006 to 2009. And these were some preliminary informations which we gave about Delhi. Delhi was not that much in news that time, and aerosol was not that famous. But now the things have changed. So these are the diagnostic parameters and isomeric ratios we generally use. So as I shared with you, interpretation of MS data is a real challenge. Like detection of molecular ion peak, what is the cluster fragmentation pattern. So if you are working on the MS data, you have to spend time on this. Then how it will help, like suppose if you are doing the bulk analysis, you are getting the spectra, right? Looking at the fragmentation pattern, you should be able to see, that's what I tell Konica also, you should be able to see, oh, it is PHS, it is alkane. And how you'll get to know? Only the NIST library will not help. Your hard work will help. Because you have to see what are the fragmentations pattern, what will be the primary ion, what will be the secondary ion. So if you are targeting any kind of this work, please be prepared to spend good time on this part of the analysis. So these are the field measurements which our students have been doing, and I think people from IIT Madras also joined us for some campaigns in Gurdaspur and Jammu. And this is the aerosol research group we have. And so as we are evolving, so we are at present collaborating with Professor Jason. Professor Jason is a student of Saintfield, and he is called Mr. Terpene. So he is, so my postdoctorate is from North Carolina, and I was very fortunate to get in touch with him in both the AAARs. So now we are trying to work on HRL CMS and try to improve and evolve ourselves in understanding this, and Konica is working on this. So maybe 1 year after 1 year, we'll be able to produce something very good. Organic aerosols and climate implications. So basically, I am going to talk about how everything is so interconnected. We are talking of chemical, biological, INP, whatever. We are talking of aerosol-cloud interactions, all is connected, and having a good knowledge of the system will help you in understanding the processes, as already said also. So when we talk of, when I am saying chemical characterization, we are doing, yes, the whole chemical characterization and source apportionment study for Jammu and Kashmir Pollution Control Board, for Jammu. But when we say chemical characterization, we are focusing on organics. And why organics? Because organic particulate matter is produced predominantly as secondary organic material, meaning by the in situ atmospheric oxidation of VOCs emitted as a primary step. So the direct and indirect radiative effects of organic PM represent some of the largest sources of uncertainty. So these are the, I gave a talk in BARC recently and they were saying you have a lot of uncertainty in your talk. So I said yes, and that is what the advantage is, and that actually opens up the area for us to do a lot of work and a lot of good work. So interactions between organic PM in water vapor through hygroscopic growth at subsaturation conditions as well as through the activation of CCN at supersaturation conditions strongly affect climate predictions and associated uncertainties. So hygroscopic growth and CCN activation are key processes, right, for accurately modeling the climate impacts of organic particulate matter. So basically what Madam was also saying, that whenever a modeler is working, modeler is looking forward for some good data from the measurement people. So this is a good team of measurement and modelers which can actually solve the process. So organic aerosol driven negative climate feedback is also very important, and so this is all the emphasis like why organic aerosols are very important. And here you can see that how the VOCs are getting produced and how it is affecting the cloud optical thickness and cloud albedo. So basically I want to say organics are very important. You also know that carbonaceous aerosols make a bulk of aerosol. In that also organic aerosol is more complex because elemental carbon we know more so and more forth. So surface active organics, again all of this, and this is a recent review which we published for the source apportionment study. Even when we say source apportionment, if you look into the details, you will see that source apportionment also has not been done in all the parts of India, and we are constrained with data. So I don't want you to get into this picture because it is very small, but it is just showing the percentage contribution of resolved chemical species in selected cities. And you can see the blue thing is OC. So OC is somehow very important, and these are the studies pertaining to the size fraction. So you can see the size segregated sampling has only been done for 6%. So it is showing the emphasis that even real-time data only 18%. And this is showing that when anybody is saying source apportionment, this I tell in the pollution control board also, when they are saying source apportionment, please ask them what composition they have done. Because sometimes people will only do WSIs, they will say source apportionment. Sometimes they will do only elemental, they will say source apportionment. But for us, the complete source apportionment is when we are doing all the composition, like all the components, WSIS, carbonaceous, because when you are working with the policy makers, it's a challenge. They will say, why you are taking 2 crore rupees, somebody is doing in 10 lakh rupees. So these type of challenges, but you should know when source apportionment is saying chemical characterizations, in what level they are achieving in that. That is very important. So broad conclusions from this review, as I just said, that most studies are focused from Delhi NCR or in IGP region. Methodology as well as source classes vary across the previous essay studies. That's why if you don't want to do any meta-analysis, you will see that the data is not available because I am doing with some other methodology, she is doing with some other methodology. So if we are talking to each other, that's why now people are trying to do this, that if we are doing some measurements in the north, we are trying to replicate them in the south. And that's why we are like making a good team with IIT Madras also. So second core research area, ice nucleating and cloud forming properties of aerosols. So I started with chemical and then we started this ice nucleating and cloud forming. So I must acknowledge the support of this group from 2017 and this is from 2022. These people have supported a lot for this because I was not working on ice nucleating particles. I was only aware of CCN because CCN was famous. Everything which is famous, which is in, we know that, right? But then I understood that no, ice nucleating particles are very important. Most precipitation in clouds initiates through the ice phase. Professor Pandithurai has already told you about INPs, that how they are important for heterogeneous nucleation phasing. In simple words, if INP is present, the freezing will take place at much warmer temperature, right? If a biological INP is there, much warmer. This is a simple statement, right? And why this is important? Because you know the lapse rate, right? Temperature is decreasing with the height. So if the freezing is taking place at much warmer temperature, that means the processes which were about to happen at some height will happen much lower, right? So this is the importance of INP, and they are rare insoluble INPs which initiate ice crystal formation. And Professor Vijay also told about the nucleation thing, so I am not showing that. So 1 in a million particle, as Professor Pandithurai said, is an INP. And this is the simple picture for INP cloud precipitation connection. Those who want to get into the details can go to this paper, Morey 2017. And so recent highlights. So when we are saying INP again, why to study INP? Because when INPs are released from biomass burning, because in India, what are the issues if you see? Aerosol is like one issue, biomass burning is another issue because of parali burning and all of that. So how to connect that with INP? So high INP biomass burning aerosols are there. Then inorganic elements lead to potentially ice active minerals through the combustion process. Similarly, when INPs are emitted in fresh BBA, so evaporation, oxidation, SOA condensation will take place, and then after the aging, what will happen? The aging enhances the ice nucleating ability of biomass burning aerosols. So from this, then glacial outwash plains are thought to be a major dust source. Glacial outwash sediments possesses higher ion ability than mineral dust. So imagine so many questions are there on INP, but if we look into the INP data, very limited data we are having. Very few groups are working on ice nucleating particles. So if we don't again have the fundamental knowledge, so going back to the chemical characterization as I was saying, so I was saying Jammu and Kashmir is not having that good resolution data. If you talk of INP, India is not having the good resolution data, right? So from that thrust area, and these are the different processes. So again, if anybody is saying that INP measurement we are doing, you should ask what type of contact freezing, immersion mode freezing, deposition mode. So like Professor Pandithurai told that he is using CFTC, right? But we are using the immersion mode measurements. So this is the study from the Himalayan region. And I will quickly summarize what we have done till now. We started first of all INP in rainwater, then in aerosols, and then in river water. I want to emphasize that these first two studies when we did analysis in North Carolina State University, and for the third one, Bruce Moffitt from Ocean Lab collaborated with us. He came with a small PCA modified thermocycler, and we did that. So we have by now we have lot of experience of having immersion mode INP measurements, how do they look and all of that. So I will just emphasize on this because I want you to understand the emphasis of INP and also what does it mean, how it is measured. So you can see we use the drop freezing assay. Wherever you will see the INP paper, you will see Wali 1971 because they give this formula to calculate INP concentration per liter. So you can see these are the sample drops at -5 degrees Celsius. So these are the 1 microliter water drop of our sample, right? And then we allow it to freeze and we go till -35. So you can see over here the difference. If you see water, it is freezing at approximately what temperature? On the x-axis you can see it is temperature, so around -20, right? But if you see the sample, this is sample from Dharamshala, you can see the freezing is happening even at warmer temperature than -10, right? So when it is happening at warmer temperature than -10, we say it is a biological INP. So this is how the INP data looks like. And this is how we calculate INP. So in the first panel, we are showing the fraction frozen. In the second panel, we are showing the INP concentration per liter water. So these are some of the results. These are the results from all over the world. And these are the results from our paper. So if you want to get into the details, please look into the paper, Yadav et al. 2019. We did a lot of good new measurements that time. We also studied time resolved, like how if the storm is progressing, how INP will change, all of that. And also we did the INP analysis of Thar Desert dust just to check like how does it look, how does it behave. And also INP activity at warm temperature is heat sensitive, suggesting a proteinaceous source. So simply till now I have told you that ice nucleating particles are very important. If the freezing is happening at warmer temperature, much warmer temperature, it is biological INP. But how you will get to know it is biological INP? The simple methodology is give a heat treatment to your sample. And if the spectra shifts like in this picture, it will suggest that yes, it will confirm that the biological INP was there. And this is a standard methodology. So we did the same, and we did also, when we found that biological INP is there, we did the plating experiments to study as Emil is doing. And then one sample showed a very unique behavior. It did not freeze even till minus 35. And I still remember Marcus was poking the stage and he was saying that there is some malfunctioning and all, but it was not there. And then we studied about the antifreeze proteins, that the same bacteria will be having ice nucleating ability also and antifreeze ability also. So that means what? Increasing the complexity, increasing the uncertainty. So it is increasing, and that's why these are the challenges, that's why we have a lot of scope to answer these questions. So then we try to find this antifreeze, however we could not find in this work, but this is one of our targets. So these are the major findings from the first paper, and then the motivation for bioaerosol research. By now you must have understood that what motivates us to study bioaerosols, because biological, INP, if it is present, the freezing will be at much warmer temperature. So, biological ice nucleators are warm INPs, like common bin are species of bacteria, fungi, algae, pollen, and the active sites of ice nucleating protein found on the outer cell membrane of bacteria feature unique hydrophilic hydrophobic patterns. So, these are the types of bioaerosols, and this is the one review which we published with Professor Sachin. So, here we emphasize that the importance of bioaerosol on crop health. This is another motivation to study bioaerosols. So this is our second study which is published in JGR Atmospheres 2022. Bioaerosol diversity in INP. So from the first work we understood that bioaerosol is very important. We collaborated with Ryan Pearl there only, and then we studied biological properties and ice nucleating properties in aerosols. And from this study some of the findings are here. And in this we did next generation sequencing. So in this what we did was, we sampled the aerosol on the filter paper. We cut the filter paper, half of the filter paper for INP and half of the filter paper for the bioaerosol research. And we did the direct DNA extraction, like the kits are there, Kaizen kits. So from there you can directly extract the DNA, and then you can do the next generation sequencing. So the benefit is that if you are doing the culture, you cannot get the complete data, but when you are doing directly from the filter, it is much advantageous, and it is one of the novel work which is happening these days in India. So these are some of the correlations of ice nucleating bacteria. Obviously when we are doing bioaerosols, we'll get to know what bacteria are present, then we'll target for what ice nucleating bacteria are present. But when we are doing bioaerosol, health implication is also one of the very important targets, right? So then we look for how what kind of disease causing genera are present and what are the other species. So when we are doing bioaerosols, we are understanding climate processes, we are understanding health implications, we are also getting some of the source traces, because some bacteria are only found in soil, some bacteria are only dwelling in cloud. So likewise we get to know the sources also. So these are some of the analysis from this second work, and then this third work was done on the Himalayan rivers with Bruce Moffett. So here also a very interesting finding we found, that you can see this is Beas, Satluj and Chenab. Please focus that here INP concentration per liter water is going till 1000, but there it is 10,000. It is not typo, right? So it is 10,000. That means the INP concentration per liter water was much higher in Chenab than in Satluj and Beas. But again, when we are saying we are giving such statements, no, we cannot give these statements on small number of samples. This is one-time study only. So somehow we have to have some measurement techniques so that we can do comprehensive study to answer such questions. So the novelty and strength of our group is offline measurements, direct extraction, filter-based sampling campaigns, and PM characterization, and method development for organic traces. Science questions are from the same theme, and the challenges are limited instrumentation facilities for INP measurement, low to very low DNA concentration. This statement look very ordinary to you, but this is a biggest challenge for us, because when I am saying I am extracting DNA from the same filter, you are having less aerosol concentration. If you want to study diurnal, imagine how much concentration you will get. Now from there you want to study DNA also. So how much DNA you will get. So sometimes, many times your experiments fail, and that's why bioaerosol research is very expensive. And that's why if you are having a good experience, it works. Also instrumentation development. So we are working, we have been working, now we are coming towards the end of this, the instrumentation development. So yeah, so this is very important, and from 2017 when I worked on the NC State facility, and finally in 2022 we were able to complete, because in between there was pandemic also. And now in Central University of Jammu we are having this facility almost functional. Why I am saying almost functional? Because we are validating the results. Because if the stage is there, we have to compare the results with the elite dust, Arizona dust, and then we'll be able to say that yes, it is fully functional and open. So this gives a very, very good opportunity for all of us sitting in the room, because you are studying any aerosol anywhere, you can do the offline characterization here, and you can get to know INP concentration per liter water and per liter air. So so this is an open hardware community ice nucleation cold stage. This because this is not the same prototype which is there visible in that picture in 2017. From there we started working on a low cost cold stage which is portable also. So this version is that, and we have submitted this manuscript in Hardware X. So I hope in the coming months you will be able to see this manuscript. I'm very thankful to this group, because I'm very thankful to this person sitting inside, and he's Marcus Peters, and he's preparing for the AppCape campaign, very famous campaign which has just started in US. And you can see the authors there, because there are contributions from RS University also. So finally I'm very happy that this facility will be functional, because I was saying last year, I was saying I'll stop going to the conferences, because I have been saying for 4 years that it is coming up. Yes?
I think you want to tell that he is the Kappa Marcus.
Yes, I think everybody knows that this is the Kappa Marcus, because he only gave the Kappa.
Famous Peterson categories.
Yes. So these are the short-term objectives of our group, which I think I have already told. And these are the long-term goals: to fill the missing link of INP and bioresource. How much time do I have? They were saying they will not give me extra.
You have 12 minutes.
To understand chemistry of processed aerosols over the Himalayas. So basically, as I said, that we are doing a lot of field measurements to understand the processes, and the upcoming projects in plant campaigns. So whenever I am saying that we are focusing on offline measurements, I never say that I undermine the importance of online measurements. But they are very expensive, but I think now we should get them soon, because the ideal case is to have the offline and online both. If we are having the offline and online combination, then nothing like that, right? So this is the Ice Crunch Indo-Suez project, which is about to start from August 1. And in this we are going to do measurements in Patnitop. And this is another project which is almost there. And then this is also approved project. In this ACRP-CRG project, I am the co-PI, where we are going to study the thermodynamic evolution of CI in the Aladar region. And talking about the Indo-Swiss, because this is very important when we are here in Munnar, because it is another going to be the high altitude site. And in Himalayan region, we are not having at present the INP measurements in the high altitude area. So this campaign is very important for us, and we are going to study again. So now what we are doing is, whatever project fundings we are applying for, we are talking with the biologist also. So here also Christy Diner is the co-PI, Zameen is the INP man, very famous, and Christy Diner will be doing the eDNA simultaneously, and I will be doing the chemical thing. And with having immersion board cold stage with us, we can do the offline measurements also. And also Zameen will be bringing all these instruments to India, like Hink, Auto. And if you want to understand what are the measurement techniques, you can go to his page and you can explore how with different types we can study INP, to study the different modes. So this is the container which we are going to use. And so this is a drop freezing picture you can see. So this is how it is captured when the drops get frozen, right? So we start from transparent ones, and then they are opaque. This tells us that the drop is freezing. So this is a picture from droplet ice nuclei count to Zurich. So, and this is another instrument which is called Hink. So I will not get into the details of the principles, but the principle of each measurement technique is different, and that's why the target is also different. So here two freezing modes can be investigated. So this is the strength of this instrument, which our immersion mode instrument won't be having. So here we can do deposition as well as condensation freezing. So the two main objectives of ice crunch are to quantify the CCN and INP properties of aerosol particles at Patni top and to delineate the contributions of natural and anthropogenic aerosols to cloud formation at Patni top in the northwestern Himalayan region. More specifically, like what are the diurnal and seasonal variability, what is the role of Indian monsoon in regional episodic events, because it's a 4-year project and we are going to settle the site there, so we are going to have long-term measurements again. Also, we are going to measure SOx, NOx, ammonia to understand the shear formation. And coming to the concluding remarks, aerosols have important implications. I think I don't have to read it. We already know that aerosols are having important implications and we need to have good team of researchers to work on this. The people from measurement and modeling side have to team up to answer these questions. And I think this workshop will help all of us in that direction because we got to know from the experts what are the directions for future research. We got to know from the participants what they are doing. So you please feel free to talk to us and to plan your research. Now I think you have a lot of opportunities to work in from Munnar to Himalayan region. So you can plan accordingly your objectives also. So before I close, as I am INEAS member, I want to share with you that INEAS membership 2024 is now open. Indian National Young Academy of Science is the first and only recognized academy for young scientists in India with a vision to promote science education and create scientific awareness. So INEAS is the voice of young scientists and provides a platform to exchange ideas, initiate discussions on scientific topics, collaborations among the new generation of scientists. So you can see over here the last date of submitting application is 15th August. So I would request you to go to the page. Okay, so the students who are in PhD won't be able to have this, but the young faculty members and researchers will be able to join this. So
Is it young by age or by aware?
it is so that is a very important question. You are not young for them. It is 40 years or less. So with this I acknowledge and thank all my students, mentors, collaborators and also the organizers. I also take this opportunity to thank all the participants for joining us for this workshop. I also take this opportunity to thank our organizing committee team. A big round of applause for them. Thank you very much.